Spherosome, also known as plant lysosome because they contain hydrolytic enzyme. Henstein first observed them and called as microsomes. It was discovered by Dengiard. Turner named them as spherosomes, and they occur only in plant cell, and some scientists call them complex microbodies. The major site of the spherosome is the synthesis and storage of lipids in the plant cell. Aileron grains, also known as plant lysosome because they contain hydrolytic enzyme. They are also known as aileroplasts because they are rich in proteins. This is the aileron layer which you get in the endosperm of cereals. Vacuoles. Sap vacuole. Outer membrane of sap vacuole is called tonoplast. Sap vacuole contains amino sugars, carotenoids, proteins, minerals, etc. Matrix of sap vacuole is called cell sap. Sap vacuole helps in osmoregulation. Anthocyanin pigment is also present in the cell sap, which of course is responsible for imparting violet, yellow or orange color. Here you can see the sap vacuole surrounded by a membrane called as tonoplast. Contractile vacuole. These are present in protists, say for example Amoeba and Paramecium. Contractile vacuole helps in water balancing and osmoregulation, and it also helps in excretion. You can see here the contractile vacuole, and the main function is osmoregulation and balancing of water. Food vacuole. These are present in protists. They store ingested food. These are also known as gastric vacuole because they are analog to alimentary canal of higher animals. Gas vacuole. These are present in cyanobacteria or photosynthetic green or purple bacteria. These are also known as pseudovacuole or air vacuole. Each gas vacuole protects from UV rays. Gas vacuole also helps in buoyancy or floating. Proteinaceous membrane is also present around the gas vacuole. Microbodies. It was discovered by Rhodin and Tolbert in plants. Peroxisomes occur in the cells of green tissue and concerned with photorespiration, which is the glycolate pathway. And you know that this photorespiration is occurring in the C3 plants. Peroxisomes are also involved in beta-oxidation of fatty acids. You can see the peroxisomes. Glyoxysomes, discovered by Rhodin, Beaver and Brindenbach. Glyoxysomes occur only in plants, especially fatty seeds, guard cells of stomata and unripe fruits. Glyoxylic acid cycle takes place in glyoxysomes. Glyoxylic acid cycle is amphibolic. That is, it may be anabolic or as well as catabolic. Due to this cycle, stomata are closed in night. Glyoxysomes contain alpha-hydroxy acid and beta-hydroxy acid enzymes. The cycle is involved in the formation of different organic acid during the ripening of fruits. This cycle converts fat into carbohydrate during the germination of fatty seeds. And it also converts organic acid into carbohydrates, which is concerned with the mechanism of closing of stomata during the night. Glyoxylate is the precursor of two amino acids, and that is glycine and serine. In addition to enzyme for glyoxylate cycle, the glyoxysomes also contain catalase and hydrogen peroxide generating glycolic oxidase. Lomosome, discovered by Murray and MacLear. These are found in fungi. These are present between the cell wall and the plasma membrane, and lomosome helps in the formation of cell wall or septum during the cell division. Centriole is also called as cell center. Each centriole consists of 9 peripheral microtubules and each microtubule is made up of 3 subtubules, which are referred as A, B, and C. Centriole is made up of peripheral triplet. Each subtubule is made up of 13 protofilaments. Each protofilament is a dimer of A and B tubulin protein. Microtubules of centriole are made up of tubulin protein. Central microtubules are absent in centriole, but in central part, hub protein is there. Of course, central tubules are absent, but it has a hub protein. Each peripheral microtubule connects with hub protein with the help of radial spoke. Terminal end of radial spoke, that is towards the periphery, forms a dense structure called X body, which is present near a subtubule. Between two X bodies, a Y body is also present. Now here you can see that peripheral having ABC and the central one lack the tubules but have a hub protein. Each Y body connected to X bodies on its both sides. AC linker connects C subtubule of a microtubule with A subtubule of another microtubule. Cilia and flagella. Cilia and flagella, they are fundamentally similar in structure. There are, of course, some differences between cilia and flagella. Both structures, they help in locomotion, excretion, secretion, and transportation. Cilia and flagella are present in eukaryotic cell except slime molds. They are also present in prokaryotic cell, exception is Anabina, and they show 9 plus 2 arrangement of microtubules. It consists of 3 parts: basal body, basal plate, and shaft. And here you can see the section of the cilia flagella. Basal body, it is also known as kinetosome or basal granule or blepharoplast. It has 9 plus 0 orientation of microtubule. Hub protein present in the proximal part of basal body but absent in the distal part. Central part of the basal granule is called central hub. Microtubules connected to central hub with the help of protein fibers called primary fibers. Secondary fibers connect the microtubules with each other. Rootlet, a conical bundle of protein fibers arising from the basal body. Rootlet have dark bands composed of ATPase. Synthesis of ATP takes place in these dark bands. Basal plate, present between basal body and shaft. It connects basal body and shaft to each other. 2 major changes occur at the region of the basal plate. Number 1, triplet of peripheral microtubule convert into doublet of microtubules. Number 2, central microtubule develop from distal end of hub protein, shaft. It consists of 2 parts, sheath and axoneme. Outer covering of cilium or flagellum is called sheath. Peripheral sheath covers the peripheral microtubule, whereas central sheath covers the central microtubule. Axoneme of shaft consists of 11 microtubules, where you have 9 peripheral and 2 central. Each peripheral microtubule consists of A and B subtubule. A subtubule has 2 bent arms. These are made up of dynein proteins. These arms directed in a clockwise direction. Arms of A tubules composed of an enzymatic protein, dynein, similar to myosin of muscles. Dynein protein have ability to split and liberate energy for movement. Cilia and flagella bend or move due to sliding of microtubules along one another while maintaining their individual length. The mechanism is very similar to the muscle contraction. Each peripheral microtubule consists of tubulin protein. Tubulin similar to actin protein of muscles. A and B subtubules connected to each other with the help of nexine protein. Both central microtubules connected to each other with protoplasmic bridge. Central sheath is present around the central microtubule. Each A subtubule linked to central sheath with the help of radial spoke. Terminal end of the radial spoke forms knob-like structure, and that is called as a spoke head. A spoke head is present in the central sheath. You can see you have the radial spoke are there. You can see the central microtubules, the peripheral microtubules, which is made of A and B subtubule. You have the dynein arms,